I'm going to talk about a very quick whistle-stop tour of 3 different studies that are related in the context of vascular dementia and mixed dementia. So neurons require a lot of, I'm going to stand on this side so I can point, energy to function properly because they have very little energy reserves of their own, and they signal to nearby blood vessels, causing them to dilate, and when they dilate, they bring in more blood. In that blood is more oxygen, glucose, and nutrients, and that nourish the neurons, and this allows them to function properly. The breakdown of this critical relationship is implicated in a variety of neurodegenerative disorders, Parkinson, stroke, ALS, and neuropsychiatric conditions. So many of us in the room don't need an introduction to dementia, but just to highlight that dementia is a bunch of different brain diseases, the most prevalent of which is Alzheimer's. Secondly, vascular dementia, tightly associated with stroke, as well as other forms of dementia. My research focuses on these two, but also the interaction between Alzheimer's and vascular dementia, mixed dementia. So just a quick recap of what Alzheimer's is. Alzheimer's is a neurodegenerative disorder characterized by the presence of beta amyloid plaques and tau, tangles, hyperphosphorylated tau, inside of neurons collectively cause the shrinkage of brain matter neurodegeneration. The vast majority of cases are sporadic, although a small are familial. And many of those sporadic factors are lifestyle factors, including hypertension, diabetes, and atherosclerosis, and up to 40% of these are modifiable. Now, I'll be highlighting the role of atherosclerosis throughout my talk, so bear that in mind. Vascular dementia, as the name suggests, is vascular. It's impairments to cerebral blood flow, can be caused by a number of different conditions. The biggest risk factor, of course, is stroke, but it can also be caused by small vessel disease, of which the biggest risk factor is atherosclerosis. So you can see the theme, that recurrent theme for this is that risk factors include atherosclerosis. And indeed, mixed dementia, or the interaction of 2 or more conditions together, comorbidity, can affect as many as 50% of the patients, although the exact prevalence is largely unknown because people don't seem to investigate this properly. Although it's probably very likely that anyone over the age of 80 that has Alzheimer's has a vascular pathology as well. So we published this review not too long ago looking at models of multimorbidity with a focus upon dementia, and unsurprisingly, we found that there were hardly any multimorbid models. So the conclusion was that we need to study and develop these comorbid models. Now, atherosclerosis, for those that don't know what that is a dynamic disease characterized by the buildup of fat in blood vessels. So very early on, you have infiltration of cholesterol molecules into the blood vessel wall that become oxidized and activated, leading to recruitment of inflammatory cells that over time cause the buildup of fatty streaks and these plaques that occlude the blood vessel lumen. And when this happens, target organs, including the brain and the heart, have a reduction in the blood flow arriving, and that can, of course, cause infarction, so myocardial infarction in the heart, or indeed in the brain, it would be a stroke. So the models we use in Sheffield include very early models of genetic Alzheimer's disease. So this mutation favors the production of amyloid beta. The atherosclerosis model is a relatively new model. It's a viral model that basically mimics the function of PCSK9, which normally would bind to the LDL receptor to prevent the reuptake of cholesterol. Normally, cholesterol would bind, get internalized, but PCSK9 prevents that uptake, leading to increased cholesterol in the blood. So that's what we're doing there. And this is just an example of a dissected aorta from these atherosclerotic mice stained for those atherosclerotic plaques. So in the neurovascular lab, we do optical imaging of cerebral blood flow, specifically cerebral blood volume, and maybe this will work. Here we go. So you have your mouse at rest, you begin a sensory stimulation, neurons become active in the sensory cortex. Through the process of neurovascular coupling, blood flows to that region, and of course, when the stimulation stops, that process stops. And we can quantify that here with an increase in HBT, which is blood volume. Oxyhemoglobin goes up and deoxyhemoglobin goes down. And then we also can stick an electrode into the brain within the active region to get simultaneous electrophysiological measures. This is multi-unit activity, which is the firing rate of neurons. And of course, post-mortem studies. So the first study I just want to quickly go over is what we published in eLife 2 years ago. And we were looking at neurovascular coupling in the atherosclerosis mouse model. And here I've just highlighted one figure. The take-home message, of course, is that there was a reduction in the blood flow in the brain of the atherosclerosis mouse model. But look at the mixed model. There doesn't seem to be any difference there. This is something we can discuss later. So this is neurovascular coupling. Along the bottom, you have the neuronal activity. Along the side, you have the blood flow. You can clearly see that the green line, which is the atherosclerosis, deviates. And for the same degree of neural activity, you're getting less of a blood flow to the brain, suggesting a functional breakdown of neurovascular coupling. A consequence of inducing atherosclerosis in the AD model was that we led to the trebling of beta amyloid plaques in the hippocampus, and there was substantial interleukin-1 mediated neuroinflammation in the brains of these animals. So just to summarize this first study, in atherosclerosis at the same age, you get the same degree of neural activity, but a reduction in the amount of blood flow, and as well as significant neuroinflammation. So thank you very much for that.